The overall goal of this protocol is to label and visualize multiciliated cells of the zebrafish in situ.
This method can help answer key questions in the developmental biology field, such as what factors regulate multiciliated cell fates in the embryonic zebrafish kidney. The main advantage of this technique is that both protein and RNA transcripts can be labeled simultaneously, which is useful in the absence of zebrafish-specific antibodies.
We first had the idea for this method when we wanted to label multiciliated cells in the pronephros while analyzing the presence of cilia after making changes to the embryo.
Begin by fixing the zebrafish embryos at 24 hours post-fertilization in 5 ml of 4% paraformaldehyde for 2 to 4 hours at room temperature without agitation. Next, wash the embryos 2 times in PBS with 0.1% tween 20 or PBST, followed by one wash with 5 ml of 100% methanol. Then incubate the embryos in 5 ml of fresh 100% ethanol for at least 20 minutes at -20 degrees Celsius. To prepare the embryos for hybridization, rehydrate the neonates in 5 ml of 50% methanol in PBST for 5 minutes, followed by 5 minutes in 5 ml of 30% methanol in PBST. Wash the embryos 2 times for 5 minutes each in 5 ml of PBST. And incubate the embryos in 5 ml of a 1 to 1000 proteinase K in PBST solution for 2 minutes.